Hey everybody, I'm going to do a series of video clips here to substitute for the class time we'll be missing on Monday, and I'll be posting these on YouTube. And I'm going to try to break things up into multiple video clips so you don't have to sit down like you're watching a movie or something and see everything at one time. Please be sure to take good notes while you're going through this, just like, because of course you take notes in class, of course, so you want to take good notes just like you do every day in class. Anyway, we were talking about viruses when we left off on Wednesday, and I had mentioned that we were going to talk about how viruses have to go through what's called a replication cycle. And remember again, viruses are not truly living things. They are particles that have genetic information, either DNA or RNA on the inside, and that DNA or RNA provides the genetic blueprints for producing new viruses. But a virus can't do that on its own. It actually has to go inside a host cell, and it will essentially hijack the host cell. The host cell's machinery will read those genetic blueprints and go through the process of making new viruses. All right, and so all viruses have to go through 5 major processes. We mentioned these on Wednesday. First, they have to attach to a host cell receptor, and that's going to be up here on the surface of the host cell and the plasma membrane. Very often that's a protein molecule on the surface. Those receptors have normal functions for cells, but the viruses hijack those for their own purposes. Different viruses use different receptors. They're very particular about their receptors, and they're not going to be able to enter a cell unless a particular cell type has their receptor that they attach to on the surface. So that's very critical. Also notice here this word virion. A virion is a mature virus particle fully assembled with its capsid if it has an envelope that has wrapped itself around the virus. If it has those little spike glycoproteins we were looking at in class on Wednesday, it'll have those. That's just another term for a mature virus particle that's capable of going off and infecting a new host cell. So that step is critical. Attachment, that's what you see here. Entry, obviously, if the virus is going to hijack the host cell, it's got to get inside the host cell. And more importantly, the genome here, the genetic material of the virus, either RNA or DNA. On this diagram, it's RNA, this diagram from your textbook. But for some viruses, that's going to be DNA. All right, and then once the genome is in the host cell, synthesis takes place. That genome has to be read. So those genetic processes we were learning about in Chapter 8, transcription, translation, the host cell is going to have to perform those. The host cell has the machinery to make that happen. The virus can't do that on its own. And then assembly is going to take place. That's what they're showing you over here. You've got to reassemble new virions, mature virus particles, and then those have to be released. That's step 5. And release generally takes place in 2 different ways. Some viruses cause the host cell to burst open. That's also known as lysis. And other viruses bud. That's what they're showing you over here on the diagram. And what that means is the new viruses push themselves through the surface. And this is how enveloped viruses get out of the host cell. Remember I said that the envelope, if a virus has that, is composed of phospholipids, and it's really a part of the cell membrane that the virus steals as it's exiting from the cell. All right, so that's kind of a generic virus replication cycle. We're going to talk more specifically about some example viruses because different viruses all have little quirks about how they accomplish those things. All right, this is a diagram from your textbook which is showing you a couple of different mechanisms by which viruses can attach and enter those first 2 steps inside a host cell. And over here on the left, what they're showing you, you've got a generic virus here. It's attaching to its receptors. Those aren't really shown, but attaching here on the surface of the host cell. And then this is engulfed. It's brought inside a membrane-covered vesicle or a membrane-covered compartment. Now, if you've studied basic biology in the past, that's a process known as endocytosis. That's a natural process by which cells bring in large molecules from the outside, and many viruses have learned how to hijack that process for their own purposes to get them inside their host cells. And then once that happens, notice the little vesicle here has to open up, and the virus breaks apart, and you release the genome. Now here they're showing you free DNA. Depending on the virus, that might be DNA or RNA. We're just looking at kind of a generic example virus here. And this is critical. Those are the genetic blueprints for the virus that the host cell will now read. All right, and then here's another different way that slightly different way that some viruses use. We've got a virus attaching to host cell receptors on the surface. And then what happens, notice the entire virus is not coming inside the host cell. This is an enveloped virus, and the envelope more or less fuses or kind of melts into the host cell's membrane. And then just that capsid, remember the capsid is a protein shell on the inside, it comes in, breaks apart, and releases the nucleic acid, which could be DNA or RNA, into the host cell. And now the host cell is going to read that nucleic acid. All right, so on Wednesday, I mentioned that viruses can be divided up into different categories based on their genomes and also the strategies that are used for replicating that DNA or RNA genome and the processes that they use for making their proteins. Viruses have proteins just like cells have proteins. The genes of viruses are blueprints for proteins of the virus. So proteins of the virus, what are those used for? The capsid shell, those glycoprotein spikes in enveloped viruses. Oops, hang on. Viruses have to make their own enzymes to carry out particular steps of their replication. And so all those are viral proteins. Those viral proteins are going to be coded for by genes on the virus's genome. So on Wednesday, I mentioned that viruses are divided up into different groups. We have RNA viruses and we have DNA viruses. And all that means in a virion, a mature viral particle that's capable of going out and finding a host cell to attach to, the genome on the inside is either going to be RNA or DNA. Let's talk about the DNA viruses over here on the right first. Many viruses are what we call double-stranded DNA viruses. And all that means is that their genomes are double-stranded DNA, just like our cells have double-stranded DNA, just like the DNA of a bacterial cell will be double-stranded DNA. And these viruses, their processes of replication, their processes by which all their new parts and so forth are made, are the most similar to cellular processes, like how our cells would operate or how a bacterial cell would operate. Which means you have genes on the double-stranded DNA. Those genes undergo transcription to make messenger RNA, and then the messenger RNA is translated to make the proteins of the virus. That little green squiggly there is supposed to be a protein, a generic protein of a virus. And just keep in mind, viruses are going to make many different proteins. It depends on the virus. Some viruses just make a few kinds of proteins. Then there are other viruses that make dozens of different kinds of proteins. It just depends on how complex they are. Viruses are highly, highly diverse. There are many different types of viruses out in the world. Okay, and then there are a few types of viruses that are called, well, they're single-stranded DNA viruses. And what that means is their genomes inside the virus, instead of the DNA being in its usual double helix, you've just got one strand of DNA. Now, when those viruses enter a host cell, you've got to replicate that DNA. You've got to make more of these. You've got to make more genomes. That's going to be true for any virus. You've got to make more of those. In order to make more of those, we've talked about DNA replication already. Okay, when DNA replication takes place, you have a DNA polymerase enzyme. Let's say we got a strand of DNA here. You got a DNA polymerase enzyme that comes along. It reads one strand, and that's called the template. And the template is telling this enzyme, DNA polymerase, how to build the new strand. So if you see a T over here, you're going to put an A over here. If you see a G on the template, you're going to put a C over there on the new DNA that you're building. So what this means is when one of these types of viruses infects a host cell, that genome, you first have to make the other strand, and then that other strand winds up being used as a template to make more of the original. Okay, and so with these single-stranded DNAs, a lot of times the single strand is referred to as a plus strand, and so the opposite or the complementary strand is going to be called the minus strand. So when those genomes originally go into a host cell, they're first converted into minuses, the complementary copy. And then these guys are read to make new pluses. You're going to go from a plus strand to a minus strand, and then the minus strands are going to be read to make lots of new plus strands over here. Also, you'll do transcription. You're going to make messenger RNA just like you would in a host, just like the host cell does. That's transcription, and then those messenger RNAs are translated to make the virus's proteins. So those kinds of viruses function pretty similar to how our cells operate. Now over on the RNA virus side, things are a little bit different. So RNA viruses can be considered to be positive stranded, negative stranded, double stranded, or then we have some that are called retroviruses. All right, so what's the difference between these types? All right, if a virus has a positive strand RNA genome or a plus strand RNA genome, you'll hear both of those terms. What that means is the genome itself can function as a messenger RNA. So when it enters the host cell is going to think it's a messenger RNA as soon as it enters the host cell. Okay, and so that means the genomes can be read to make proteins directly. And again, these virus proteins are essential for making new virions or virus particles. Proteins play structural roles in new viruses. Also, to replicate the RNA, this is going to function in kind of a similar way to DNA replication. If these are the genomes you've got in the virus, where they're called plus strand RNA genomes, the enzyme that is going to copy or replicate the RNA first has to make a complementary negative strand. And again, that means if you've got an A on the plus strand, the enzyme is going to put a U in the new negative strand it's making. If it sees a G on the plus strand, it's going to put a C in the new negative strand that it's making. All right, and then these are read by the enzyme to make lots and lots and lots of new plus strand RNA genomes. The reason for that is because those are the ones, the plus strands are the ones that wind up inside the virus particles. So you got to make a lot of those. All right, like they're showing you over here, in order to make a new virus particle, you got to have the virus's proteins, and you got to have lots of these genomes to package up inside there. Okay, so what's a negative strand or a minus strand RNA virus? Plus strand, again, just means that's an RNA that the host cell is going to treat like a messenger RNA. So a ribosome is going to attach and do translation, make a protein from it or proteins from it. So viruses that have negative strand RNA genomes, when their genomes go inside the host cell doesn't immediately recognize those as messenger RNAs. Instead, they have to be converted first to plus strand RNAs. Okay, and the plus strand RNAs, and they're showing you more of them over here, those are going to be the ones that are translated to make proteins. And also those plus strand RNAs are going to be the templates that the RNA polymerase enzyme, remember RNA polymerases are the enzymes that build RNA molecules. So anytime you're building RNA, an RNA polymerase enzyme is going to perform that. So these guys, the new pluses, are going to be read by RNA polymerase to make lots of new minus strand RNA genomes. And then once you've got all these virus proteins, you've got these minus strand RNA genomes, you can assemble your new virus particles. And one thing I want to point out here as well, something to think about, our cells have RNA polymerase enzymes. We've talked about those already because those are the enzymes that do transcription. They build messenger RNAs, and they're also responsible for building the tRNAs and the ribosomal RNAs and those regulatory RNAs that your textbook was mentioning that we didn't really talk much about. But let's think about it. Our RNA polymerases read DNA and build RNA from it. These two types of viruses, they have their own RNA polymerase enzymes, and they read RNA and build new RNA molecules from it. So they're going from RNA to RNA instead of from DNA to RNA. Why is that important? That's a good thing when we're trying to figure out ways of coming up with new drugs to treat viral infections with, because if you could come up with a drug that screws up these kinds of enzymes but not these kinds of enzymes, that's probably going to be a good drug, because hopefully it won't harm our cells, but it will screw up the virus's enzymes that it's trying to use to replicate itself. All right, another category down here, there are some kinds of viruses that are called double-stranded RNA viruses. That means over here, this is inside the virus particle, you have both a positive and a negative strand of RNA. They're bonded together, kind of like a double-stranded DNA would. And when that enters the host cell, you've got to make plus strands right here, because those are going to be your mRNAs. Those get read to make the virus's proteins. You've also got to make more of those double-stranded RNAs. Again, you're going to have an RNA polymerase enzyme that does this. And once you've made lots of virus proteins and you've made all these new genomes, you can assemble your new viruses over here. All right, and a final example, the retroviruses. And an important example of a retrovirus is HIV, human immunodeficiency virus. And these are different. Inside the virion of HIV, you have a plus strand RNA genome. It's RNA. Once that enters the host cell, it gets converted first into a single-stranded DNA copy and then into a double-stranded DNA copy. So these viruses are called retroviruses because their RNA genomes get converted into double-stranded DNA. So it's backwards from what we've learned about with regard to transcription. Transcription, you start with DNA and you make an RNA copy of DNA. These viruses have single-stranded RNA genomes, and when they enter their host cells, they get converted into double-stranded DNA. So because of that, they're called retroviruses, because they're going backwards. That process is called reverse transcription. All right, so that's something that the retroviruses do that typical host cells don't do. This is a process that's largely restricted to these types of viruses. Now, once those HIV or other retroviruses, once their genomes have been converted to double-stranded DNA, then the host cell treats it like its own DNA. So you do transcription, you make messenger RNAs, and those get translated into the virus's proteins. You package up more of those RNA molecules, those RNA genomes, over here into the new viruses that you're making. And we'll talk a little bit more about HIV shortly. Okay, a little bit more about virus replication. Again, the genome of the virus does have genes on it. The number of genes varies according to the type of virus. Some viruses just have a few genes that they need, and some viruses will have over 100. It depends on the type. Those genes code for the virus's proteins. Those proteins in general can be divided up into two categories. Structural, that means they're part of the virus particle itself, and those proteins will make up the capsids, the protein shells that protect the genome. If you have an enveloped virus, those glycoprotein spikes in the viral envelope. Some viruses have a layer in between. Oops. I'm never going to stop forgetting that my pen's not turned on. Some viruses have a middle layer called the matrix. Remember matrix, you learn about that when you study tissues. It's kind of like filler space between cells. In this case, the matrix of a virus is where you have a filler layer in between the capsid and an envelope, and an enveloped virus. So if a virus has a matrix, just keep in mind there's lots of diversity with viruses. It'll be built from matrix proteins. Replication proteins are another category. So these are the enzymes and other proteins that might be needed to make new copies of the genome of the virus. So for many viruses, they just hijack the host cell. The host cell does all this copying for them. But for many others, they have to provide the enzymes and machinery to copy those viral genomes, to make new RNA genomes or to make new DNA genomes. And again, all right, so these, we don't have a lot of good drugs for treating viral infections. And one of the, the key reason for that is it's hard to come up with a drug that is going to screw up the virus in some way while not also harming the host cell, because these viruses are hijacking the host cell to copy themselves. So a good antiviral drug should home in on an enzyme or some other process of the virus that the host cell does not need for its own survival. All right, so again, like we were talking about, those RNA polymerases of the RNA viruses, those enzymes read RNA templates and build new RNA molecules. Our RNA polymerase enzymes don't do that. They read DNA and make RNA from that. So if we can come up, there's a lot of research into, can we come up with drugs that will screw up those viral RNA polymerase enzymes without harming our own enzymes. Okay, once you have synthesized, you have built all of these new virus particles, they have to get out of the host cell. Now, as I mentioned earlier, some viruses get out by lysis. They actually cause the host cell to die, and it falls apart, breaks apart, and then the new viruses get released. Enveloped viruses get out through budding, and many of those bud at the cell membrane, the plasma membrane at the surface. And this is a diagram from your textbook, which is showing you an example. So this little squiggly thing here that looks like a yellow worm, that is actually an RNA genome of this particular virus. And the little purple things there that are surrounding the RNA, those are the capsid proteins. So that's that protein shell that's surrounding the virus. Remember, viruses have lots of different shapes. So this one kind of has a spiral shape or helical shape to it. And then notice that that capsid with the RNA in it travels up here to the surface, and at the surface you have the virus's glycoprotein spikes. They're already sitting up there in the cell membrane. And the reason for that is because the host cell treats these viral spikes like they're some kind of membrane protein of the host cell. The host cell just thinks, oh, this is like some transport protein I've got to send up to the host cell surface. So they're all sitting up there waiting for the capsid to approach. The capsid approaches, as you see there, and as that happens, the capsid proteins and the spikes are very strongly attracted to each other, and so the capsid pushes itself through the host cell membrane, and the spikes wrap themselves around the capsid, and as that happens, a piece of the membrane pinches off as well, and that becomes the virus's envelope. So you can see that over here. Over here, the virus has pinched itself off. It's stolen part of the host cell membrane that's become the virus's envelope, and now you've got those little glycoprotein spikes there in the surface. Remember, those spikes are often important for the virus to attach to another host cell. So on the left, you've got a diagram of that happening, and over here, this is a colorized electron microscopic image of some actual real viruses budding out of a host cell. All right, I'm going to stop this particular video clip there, so you can take a break if you want to. On the next one, I'm going to talk about some example types of viruses and talk about the different steps. There's some little quirks from virus to virus in terms of how they achieve the steps of their life cycle. So that'll be the topic on the next video clip.